back to our YouTube channel. This is Felix Ruben Omondi, and today specifically I want to talk about viral replication. So viral replication, we are going to look into HIV life cycle, and this is really going to help you understand the reason why sometimes a patient will take PEP, post-exposure, and then the PEP helps the patient before the patient gets the virus within a period of 72 hours. Now, these are some of the things that you need to understand in this process. Number one, we are going to deal with CD4 cells. In the whole process, CD4 is just a collection of cells and substances that make the immunity of a cell. That is what helps the body in fighting germs and many microorganisms that are foreign. Now, another thing that you must understand also is host cell. The host cell here is the CD4, and this is what the HIV virus targets so much. Now, somebody would ask you, what is a virus? A virus is some type of a pathological microscopic substance that is in genetical formulation. Now, the genetical formulation in a virus of HIV is the RNA, which is the ribonucleic acid. Now, something that you need to understand is that HIV is mostly dealing with the RNA. And the virus of HIV is basically the RNA, and it is what normally comes with single strand. So the single-stranded RNA is our main basis over here. Now, the DNA has two strands, and most of the stuff into the DNA helps to pass information to another generation, the genetical information to another generation. Now, that is the basis, and that is what makes the difference between RNA and DNA. Human body, we have the DNA, and most organisms, but the virus has what we call the RNA strands. Now, we are going to understand the process of 7 steps of viral replication. Number one, we are going to have an acronym that is going to help us understand all this process from step 1 up to step 7. Now, we have what we call the HAFRITAB. HAFRITAB is HAFRITAB. The first one stands for attachment. The second F stands for fusion. The third step stands for reverse transcription. I here stands for integration. Then T here stands for transcription. A here stands for assembly. B here stands for budding. Now, this is the steps. This is an acronym that is going to help you understand what the life cycle here means. Now, I'm going to explain from each step, from point 1 up to point B. Now, into the attachment, the host cell, that is the CD4 cell here, is busy with receiving and sending information. The information here is the virus. So it receives the information, and the host cell receives the information, and the CD4 opens the entry of viral access. That is where the replication starts from. And HIV virus is, it's called retro. It simply means it replicates going backwards. Now, once it has gained access into the blood system, we have what we call fusion. Now, into the fusion, the message has been sent successfully, okay, into the CD4. And the host cell has got the nucleus. The information has been sent here successfully, and the contents that are being deposited into the host cell is what we call the HIV RNA. So the HIV RNA is being dumped into the host cell. Now, into the reverse transcription, we are going to use an enzyme called reverse transcriptase. It is just going to help us make another strand. The RNA HIV, which we understand is one strand, it attaches to the host cell and makes another double strand that is called the viral DNA. So the viral DNA is being made here, and now that gives us what we call the double strand DNA. Now, we are going to have what we call integration. Now, into the integration process, we will understand that the HIV DNA combines with the body system to make what we call DNA. It makes what we call the DNA of the body. Now, the DNA of the body, we are going to make a new HIV system that is going to be reciprocated or replicated into other forms of HIV into the last step that is called budding. Now, before we go to budding, into the assembly, this is where now the transcription, this is where an information of making new HIV is formed. So information of packages are being made at this point in long strands of chain. And mark you this into the edges of CD4, the virus is having some kind of fatty tissues. So information is being made here, and the same information that has been made here is what helps us to form new HIV into the assembly stage. So it is like something has been manufactured using an information that has been brought in from the previous step that was transcription to make something that is new. Now that is going to make us have what we call a package. Now, into the budding, the newly formed HIV access the cell wall of the CD4, and then it pushes itself outside to another cell, and that's how another one is formed, another HIV is formed, and it reciprocates until it is viral into the whole body. Now, somebody would ask you, what is the relationship between CD4 and viral load? When CD4 is high, the viral load simply means it will go down because there is high chances of fighting the virus. Now, this is a patient that is suppressed and a patient that is adhering very well to drugs. And now when this CD4 level is going down, it simply means that the viral load is shooting high, and several opportunistic infection invades into the body because they take advantage of the new formulation that has been made, which means the system is very weak and somebody will be able to go to another stage. So that is how you are going to understand the whole life cycle of HIV. Thank you for watching this YouTube channel. Subscribe, like, and share. Thank you. Bye.